Hey everybody, we're working on chapter 12, cardiovascular system physiology. So we've moved on from the endocrine system and we're focusing on the cardiovascular system. Again, nothing works in isolation. So as we progress through this PowerPoint, you're going to see some references to endocrine material. You may see some references to skeletal material, etc. And it may also reference some future systems that we're going to be dealing with. The circulatory system, as you learned in anatomy 101 or 102, can't remember which one that was, consists of the heart, of the blood, and the vessels that are part of a closed system that are designed to move oxygen, remove CO2, and move nutrients and water content throughout your body to the various cells that need it, and to allow for things that your body doesn't need to be excreted by the urinary system. The entire process is controlled by the nervous system and mediated by the nervous system because there is a whole electrical stimulation part of the heart that allows for pumping of the blood to do the movements that it needs to do. And throughout the system, there are also other nervous system interventions. The endocrine system is involved simply because hormones travel through the blood to get from their initial gland or organ to their target cell. So let's focus for a minute on something that is going to be one of the most important diagnostic tools that is used by the medical field, and that is blood. It should make sense to you that blood can show you a lot of different things about your health because of what it contains. So the hematocrit version or part of, let me get my little laser pointer here. This part of the blood, if you were to take a vial of blood, this blood has been placed in a centrifuge so that the different layers separate. So what you see when it comes out is a homogeneous mixture. You don't see these individual layers. But when the vial of blood is placed in a centrifuge and spun at a very, very high rate, the portions separate based on their densities. Okay, so the least dense material is going to be on the top, the most dense material is going to be on the bottom, and the layers in between are somewhere in between in density. So let's take a look at each layer. What's referred to as hematocrit is the red portion of the overall blood draw. It consists of red blood cells and polymorphonuclear cells. So poly means many, morphonuclear refers to the nuclei within the cell, and that it's morphed nucleus. Eosinophils and neutrophils are white blood cells that are examples of those polymorphonuclear cells. So you have red blood cells. These are examples, an eosinophil and a neutrophil are examples of those polymorphonuclear cells, and you can see what they mean then. So the dark purple in there in both of them is the nucleus that contains the DNA material. Just a little side note, red blood cells do not, believe it or not, have a nuclei. They do not contain a nuclei because they do not need to make new proteins, which is what DNA is for. So they do not need to make new proteins because they only, quote unquote, live for about 120 days and then they're destroyed. So of an entire blood draw, 45% is hematocrit, or these red blood cells and these white blood cells, okay? Morphonuclear white blood cells. 1% is platelets, and other white blood cells like lymphocytes and monocytes. So the platelets are represented by the little stars, the lymphocytes and the monocytes, and other mononuclear, singular, more recognizable nucleus cells are shown here. And that is 1% total of your blood draw. 55%, or the highest amount of your blood draw, 55% is plasma. And plasma is the liquid portion. It turns out as a buff yellow coat, or a yellow color. It contains water, proteins, and electrolytes. So hopefully you're aware, because of your study of the musculoskeletal system, that sodium, chlorine, potassium, those are your electrolytes, and they are necessary for a lot of different things, for starting and stopping processes, for helping in nervous system and skeletal cell connections. So those are carried by your blood. All different types of proteins, thousands upon thousands of different types of proteins. That would include a peptide hormone, because remember, a peptide hormone is made of a bunch of amino acids. Well, that's what a protein is. They're not folded in the same way, but enzymes are proteins, so any enzymes that are found in your blood, and then the water component of it. Just to get you oriented, red blood cells are going to be referred to as erythrocytes, and white blood cells are going to be referred to collectively as leukocytes. So you are a phlebotomist and you take a blood draw, and immediately the body begins the blood clotting process. And there's a couple of things in this picture that you should recognize from that previous picture, and a couple of things that are kind of new. So when you stick a needle into a vessel, you create an opening in the vessel wall. So here's our vessel wall. You create an opening, and that opening is going to release the contents of the blood vessel. Well, again, if we think back to the last slide, that's going to be red blood cells, maybe some leukocytes, it's going to be water, it's going to be proteins, it's going to be whatever is in the blood at that time. If it is a microscopic break, okay, you're not going to bleed out. And that's what we're talking about here when we're talking about taking a blood draw and the clotting process that follows. So this process involves many enzymes in starting the process or initiating it, and then maintaining the process until a clot is developed. What I want you to recognize here is that nowhere in this description, nowhere here, do you see the word erythrocyte, okay? And yet, when you look at the pictures, this blood clot involves red blood cells, right? The clot itself may have red blood cells in it, but red blood cells are not called to the site by clotting factors to get that blood clot formed, okay? So I want to be very clear that erythrocytes are not a part of the process of the blood clot, although they do get entangled within the fibrin strands of the blood clot. Okay, so clotting factors are going to act upon a substance called prothrombin. When they do that, okay, so this break initiates that process, a bunch of different clotting factors to be released. They're added to a protein that is in the blood to turn it into thrombin. That is a signal to fibrinogen, another protein, to turn into fibrin. Okay, what's the difference between the two? One of them is soluble in water, okay, can be dissolved in water. That means it's floating in the blood. and one of them is not. And we do not want this form of the protein to be able to be dissolved by the blood beneath it, or the clot will never develop. So essentially, injury triggers the release of clotting factors, vasoconstriction limits the blood flow, and platelets form a sticky plug. So your platelets, these yellow things, are already in the blood. They form a sticky plug with some of these red blood cells until the fibrogen can be turned into fibrin. Then we start to get these light blue, it's almost like a net that comes in here and fills in the spots and develops the clot, so that an insoluble clot, so that no more blood could leak. So at this stage, you're going to have blood leakage. So free blood flow, okay, from your arm, from your blood draw, reduced blood flow, okay, maybe leaky, and then complete clotting so that you have no more release of blood. So we think about, okay, why is the blood clotting process so important? And it's obviously because you have to have 3 to 5 liters of blood in your body to be able to maintain all the processes that require the parts of the blood and the system itself, the cardiovascular system that it travels in, to be able to get jobs done around your body and be responsive to the environment. So now let's take a look at the plasma portion, the 55% of the blood that is made of water, proteins, and electrolytes. Of that 55% of your blood, 90% of that layer is going to be water. Again, you're going to have charged particles that are necessary for a lot of different processes in your body. You're going to have glucose that will be sent to the cells to be broken down during cellular respiration for energy. You're going to have amino acids from your food, your digestive system, and vitamins, both of which are necessary for the protein building process. And remember, proteins are used for building structures all over your body. You're going to have urea, bilirubin, and creatinine, all 3 chemicals in your blood that are waste products from other processes in your body involving the liver, involving the kidneys, involving adrenal glands, involving other organs. You're going to have oxygen and CO2 in the plasma. You're going to have hormones, any of the 3 types, steroid, amine, or peptide. You're going to have proteins that are made in the liver, which include albumins, globulins, and fibrinogens. Remember, in the previous slide, we saw the word fibrinogens, so this one is responsible for blood clotting. These have other energy benefits. And when we remove the clotting proteins from plasma, it provides us with what we call serum, plasma serum. Okay, sometimes we want to do that because we want to focus on the other components to determine health conditions, and so we'll remove all those clotting proteins so we can keep this material in a watery form, or a liquid form instead of a clotted form, a liquid form, so that we can manipulate it as we need to in the lab. So that's another huge component of your blood. The other huge component of your blood that was located in 2 different areas are your leukocytes. And this is just a little chart that kind of shows you how all the different leukocytes are made. Do I want you to memorize all of it? No, I don't, but I do want you to understand how the chart works. So in the beginning, okay, when you're making white blood cells during development, um, you have stem cells, which are called hematopoietic stem cells. They are uncommitted. So they're a cell that has multiple parts, multiple organelles, and with certain chemical signals, they will turn into other things. Okay? So the first step is a chemical signal is given to this multipotent uncommitted stem cell. That particular chemical produces blast cells. So the blast cells are a little bit more detailed. They're smaller. You notice that they have a little bit more cell-like structure. This almost looks like a blob. This looks a little bit more like the cells that we're used to. If another chemical is added to this blast cell, it's going to eventually turn into a red blood cell. Okay? If you take a red blood cell and you add a different chemical signal, because chemical signaling, attachment to receptors on the outside or intracellularly or nuclearly, is what is going to change this basic blast cell, in this case, into a megakaryocyte. This is a precursor to platelets. So these guys are going to hang around until platelets are needed or need to be replaced in the bloodstream to maintain the percentage of platelet count that is healthy for a body to respond to any blood clotting issues. Because remember, you have internal blood cuts or damage as well that need to be fixed. Things that you wouldn't even know were happening because they're not bleeding to the external surface. So that megakaryocyte then, add a chemical to it and it's eventually going to make a platelet. Okay? Take another blast cell, add a chemical to it, add another chemical to it, and you're going to get a monocyte. And those will eventually be made into macrophages. And macrophages, I always think of them as Pac-Man. They're the ones that eat large invaders at tissue break sites, so that you're not getting bacteria, large viruses. They're just coming in and they're chomping down on them and dissolving them so that you don't get an infection. Add a different chemical and you're eventually going to get a neutrophil. So you get the idea. You add a different chemical to a blast cell, eosinophil, a basionophil, a B cell, or a T cell. Okay? So here you're going to take this original stem cell, add a chemical to it, and it's going to create some cells that hang out in the bone marrow that are just waiting for someone or some chemical to come in and tell them, hey, we've got a serious infection here, we've got cancer, we've got mitosis gone crazy, which is cancer, we've got something going on here, and we need to produce the stealth bombers of your body, of your immune system. And so these are immune cells that are called upon when you have an infection that needs to be taken care of pretty quickly. So if we focus on this left-hand side, the name of this is erythropoiesis, and it's the creation of red blood cells from a cell that's hanging out in the bone marrow again, that's hanging out in the bone marrow, waiting to be turned into a red blood cell. So remember when I said 120 days, you got to replace some red blood cells. They no longer are capable of making that trip around the body over and over and over again. They're getting worn out. Their parts are unable to carry the gases the way they need to, and so they're going to be destroyed by the spleen. So what did I tell you? I told you that we were going to talk about the endocrine system. Here it is. Okay, so a hormone from the kidney is going to stimulate this process in the bone marrow. Okay, so what is the bone marrow? Is the bone marrow the original organ or is it the target? What is the kidney? Is it the original organ, endocrine organ, or is it the target? The original makes the hormone, the target does something because that hormone is present. And the something that it's going to do in this case is create a red blood cell to replace old, dead, worn out um, blood loss, replacing blood that's lost due to an injury. So what's going to stimulate that? Well, we said erythropoietin secretion from the kidneys is in response to a decreased oxygen level in the blood that's delivered to the kidneys. So the clue here is, or the stimulus, which is always in green, the stimulus that says, hey, wait a minute, we don't have as many red blood cells as we need, is that there's not as much oxygen being carried in the blood. And that should make sense. If the red blood cells are no longer capable of carrying oxygen, then it's probably because they're broken down and the hemoglobin can no longer hold on to the oxygen and take it to the cells. So the kidneys pick up on that. They give off their hormone. The hormone travels through the blood. That's what these orange ones always are. Travels through the blood, so it increases the level of that hormone in the blood. It reaches the bone marrow, and it causes an increased production of those red blood cells. There's an increased blood HB concentration. So HB is the hemoglobin that is attached to those new blood cells, okay? They now have gone up. The blood oxygen carrying capacity goes up, and you have restoration of oxygen delivery. So positive or negative feedback? This is actually negative feedback, because when we have a stimulus that goes out of the norm and we're trying to restore it, that's a negative feedback. Now, if we had something go out of the norm and we wanted it to magnify and go even further from that norm, that would be a positive feedback mechanism. But that's not what the kidneys and the bone marrow are responsible for doing in the case of erythropoiesis. So we've talked about before that processes require, it's kind of like a recipe, processes require multiple ingredients. And besides the hormone that's going to stimulate the red blood cells to be made from the bone marrow, those precursor cells that are in the bone marrow, we have to have iron, we have to have an excess of folic acid, we have to have vitamin B. Why is that? Because when we're creating a new cell, no matter what it is, when we're creating a new cell, in the case of red blood cells, we need the folic acid, so we have normal cell division from those precursors. So we have hemoglobin, which is a protein that, a couple of different proteins that hang off the red blood cell, where the oxygen attaches. We need to be able to make that, and one of the ingredients for making hemoglobin as a protein is iron, okay? Folic acid for helping to support the process of cell division when those cells are made from the precursor stem cells in the blood marrow. And then to get the folic acid to work, you have to have vitamin B12. And so that's going to be kind of an aid in getting this to work, which will get this to work, which will allow those new red blood cells to be made. So I always think of it as a recipe. You know, we say this, and this has to happen for erythrocytes to be made. Well, that is true, but a lot of times we're leaving out the support systems, the support ingredients that are necessary to get the process finalized. So what are some things that make erythrocytes different than other cells? Their main function is to carry oxygen. They are biconcave discs. I think they look like a lifesaver that hasn't had the middle popped out. They're essentially bags of hemoglobin, which is, as I said before, a specialized protein. They have very few organelles. They do not have a nucleus. They outnumber white blood cells or leukocytes 1,000 to 1 in your bloodstream, hence the red color of your blood. They contain they contain a plasma membrane protein that is for targeting and for recognition, and they are a major factor that contributes to blood viscosity. So I don't know if you've remembered from past physical science classes, but viscosity refers to the thickness or thinness of a fluid, and that could be air. I know you probably don't think about it this way, but air is actually a fluid because it flows from one place to another. And you know that because of wind. And then all liquids are fluids because they flow from one place to another. Well, if we're looking at honey as compared to milk, they have different viscosities, and that's different thicknesses. Their ability to flow then is from one place to another is going to be determined by their viscosity. Well, because your blood is in an enclosed system of blood vessels, it is important for it to be able to flow through those tubes and go out into the body, return to the heart, return to the lungs to be oxygenated, and return back to the heart to be returned to the body. So that's a lot of movement. If your blood is very thick, it's going to be difficult for it to move, and it's going to make the work of your heart harder to be able to pump it and to get it to move through those closed portals. So as I said before, the RBCs only live 120 days. They are sent to the spleen. They're targeted by chemicals, and they're sent to the spleen to filter them out of the blood. And then they get sent to the liver to break them down. The iron is stored by the liver and sent to the blood marrow so that it can be used to make new RBCs. And transferrin is the protein that takes it to the bone marrow to be used when RBCs are made. Okay, so when they're no longer viable, can't use them anymore in about 120 days, they're all worn out, they need to be retired. The spleen, the liver, and the bone marrow are 3 areas that are involved in recycling them. Let's put it that way. So let's look at 2 red blood cell or erythrocyte disorders. First of all, the sickle cell disease. Sickle cell disease affects mainly African Americans. It's not exclusive, but there is a very high incident rate in African Americans, and it is a genetic disorder. It's where, if you're looking at the normal biconcave red blood cells here, people with sickle cell disease, the genetic deformity that they have on their red blood cells is that they do not make the hemoglobin that makes that biconcave shape correctly. And there's 2 problems with that, maybe 3. They look weird, okay? They block When these all get caught up together, they'll block small capillaries and cause tremendous amounts of pain, hemorrhage, local hemorrhage into tissues, so you get hematomas, and tissue damage as a result because of their shape. So genetically, the gene that controls the normal shape of a red blood cell is modified or changed in people with sickle cell, and they end up with some regular shape and some odd shaped cells. A second disorder is anemia. There are a lot of different types of anemia, but anemia is essentially an abnormally low red blood cell count, as you can see in the picture. One of the best ways that you're going to know that anemia is present is a reduced oxygen carrying capacity. There's other reasons for having low oxygen in your blood, but one reason is because you have fewer RBCs. So when you're doing that blood draw, you're going to look at a couple of different things to indicate whether you're looking at an anemia issue, a blood issue, an actual component of the blood not being present, or if you're looking at a respiratory cause for low oxygen level in the blood. So what are causes of anemia? Dietary deficiencies in iron, B12, or folic acid. That should ring a bell with you because all three of those things are necessary for making new red blood cells. So if you're not getting them in your diet, then you're not going to be able to make new ones, and you're going to have a low level of them. Bone marrow failure due to toxic drugs or cancer. Okay, so it can be a side effect of treatment for cancer because the bone marrow is very susceptible to toxic chemicals that are given during the treatment of cancer. And if the bone marrow fails, since that's where erythrocytes are made, you're going to have an issue with making new ones. Direct blood loss from the body, hemorrhage, again, that can be internal as well as external. External is going to be obvious. You're not going to have to do any weird diagnoses there. It's going to be obvious that there's a low red blood cell count for a person who's hemorrhaging externally. But internally, if you're not you know that all of the symptoms of anemia, fatigue, and some other things that are associated with it are showing up, and you don't have a way to see whether they have blood loss on the outside, you're going to have to start to look for internal hemorrhaging if all of these other things are not true. Inadequate secretion of the hormone necessary, which is often a symptom of kidney disease. Again, this is necessary to stimulate the bone marrow, to make the erythrocytes along with all of their other ingredients. So you can start to see a process here. If any one of these things are affected, then you're going to have a reduced amount of oxygen carrying RBCs. And that's kind of a cascade effect. Should be obvious when you're thinking about that, because every cell in your body requires oxygen to be able to go through cellular respiration. And if you don't go through cellular respiration, you don't have ATP energy for your cells to be able to do their unique and individual job. So, big deal. All right, so now let's focus a little bit on the leukocytes. So we're leaving the erythrocytes, and all I'm going to say about these is just to show you some difference. I think they are so flippin' cool looking. Like, I love this colored picture of it in particular. But here you can see like the polymorphic versus a regular old nucleus. Now, this is a regular old nucleus. It looks kind of weird, but it's a regular old nucleus. But you can see a real difference there in the nuclei of the eosinophil and the neutrophil. So what are their jobs? Neutrophils, I mean, you can look through all of these, and it all has to do with the immune system. It all has to do with keeping your blood, quote unquote, clean of parasitic invaders, trying to minimize infection, and then having a kind of a memory. Some of these are going to be involved in memory for when that invader comes into the body again. You're going to have a memory of, oh, got to get rid of it. It's going to cause an infection. We recognize it, target it, and kill it, so that you're not, and that's the T and B cells, so that you're not going to have to worry about getting reinfected by something that your body already recognizes. So erythrocytes' primary function, or really, really important function, is to carry oxygen to all the cells. And then a generalized summary of white blood cells are that they have to do with the immune system and protecting you from invaders from the outside world, whether parasitic or bacterial or viral, just all the pathogens that can cause issues for you. So I talked about the fact that on the first slide that the heart is involved, the blood, as we've been talking about, is involved in the cardiovascular system, and then all of the vascular portion of it, the vessels, okay? And so what we're going to take a look at in these next few slides are the different types of circulation, okay? And I said in lab the other day, this is the plumber cardiologist. This is the specialist who is worried about blockages, who is worried about diffusion, who is worried about blood content, blood viscosity, blood movement, blood concentration, blood filtration, because as you can see, this is a closed system and you want it that way, because when we say we bleed out, what we're talking about is some sort of opening anywhere in this system, at the capillary level, which would be in your skin, to the deep veins and the deep arteries, to, God forbid, the heart, and to all of the capillaries in your lungs and going away from and to your lungs. So if there's an opening in any of those, all of the blood that's involved in this closed system can be pumped out that opening over time. And with that blood loss, you're no longer getting blood to the skin through the capillaries, to the lungs for oxygenation and release of CO2, etc., and to the nervous system for that matter. So the systemic part that the plumber cardiologist deals with in circulatory problems of the cardiovascular system, the systemic circulation is the arterioles, the capillaries, the venules, so small veins, small arteries, capillary beds where CO2 and O2 is being directly exchanged. So that's right in these, okay, where you've got oxygenated blood coming in to these capillary beds, and all the cells that surround it take in the oxygen that that blood has to give, and in the meantime they give off the carbon dioxide that's a waste product from cellular respiration. So once they give that off, those cells continue on through the deoxygenated side of the cardiac system, or the right side of the heart, and return it to the heart to be oxygenated. Okay, so the systemic part of it then is where in your body there is interaction at the tissue level to exchange blood gases. Excuse me. The pulmonary circulation, kind of obvious. You're exchanging blood gases, but this time you're taking blood from the heart, deoxygenated blood from the heart that's carrying CO2, and it's going to go to capillary beds in your lungs. It's going to give off the CO2. You're going to breathe that out, and it's going to um, give it oxygen from the alveolar sacs in your respiratory system. It's going to give the blood oxygen, and then that blood goes back to the heart and then gets pumped out, and it goes back to the systemic circulation for that process to return again. Okay? Closed system. It's pulmonary. Microcirculation discusses, so it's going to kind of detail then what I was just talking about is happening in the capillary beds. Okay, so when blood flows into the systemic system, the oxygen pressure of the hemoglobin, that protein on the red blood cell, is greater than the oxygen pressure in the interstitial fluid. So this is going to cause, we move from high to low, always. That's what diffusion is. So this is going to cause oxygen to flow from the hemoglobin to the interstitial space. Okay? To get ready for that carbon dioxide and oxygen exchange in the capillaries. So pressure differences are what is causing the movement of those gases. The CO2, so if we're staying still in these capillaries, the CO2 pressure in the interstitial fluid is higher in systemic circulation than it is in the blood cells that just pass through into the veins. So CO2 moves from the interstitial space into the veins, attaches to the red blood cells, which then take them back to the heart, back to the lungs. Same process happens up here, but instead of being interstitial space, it is alveoli sacs. But this time we're dumping off CO2 because the CO2 pressure in the alveoli sac is lower than the CO2 pressure in the red blood cells. So they dump off their CO2. You know what? I can do a little arrowing on this, I think. So CO2 is higher here. It's going to go out into the alveoli. Okay, so that's the CO2. It's going to be dumped. And then O2 from the alveoli sac that you just breathed in from the air is going to come in to that and then travel back. Atrioventricle. And then you know the rest of the story. I'm not very good. There we go. Okay, so down here, we're talking about O2 going out. And CO2 coming in to the cells. Okay, so just the opposite. All right, so gas exchange. So microcirculation is what we talk about when we talk specifically about the exchange of gases at the capillary level. So I included this so that you could maybe use it. I don't know if you want to take a screenshot of it or something, but I would like for you to think about following the path of a blood cell and the gas it carries, starting at, it's saying to start at L. So we're going to start here. We're going to start there. And we're going to follow where the blood goes, okay? And as we do it, we want to talk about what gas does it have in it, okay? So this is for you to use if you choose, if you want to take a screenshot of it or something, just something that you can, you know, use to practice with. But here is how the blood travels. So if we start at the pulmonary trunk, okay, which is where the arteries and the veins are going to go either into the lungs or out of the lungs. If we start at the pulmonary trunk, it's going to branch into arteries, arteries into arterioles, arterioles into capillaries within the lung, pulmonary venules, and the capillaries. That's where the micro circulation is happening, where the CO2 and O2 exchange is happening, to the pulmonary veins. Pulmonary veins are going to dump off CO2-laden cells into the left atrium. They're going to pass through the left AV valve into the left ventricle. They're going to go through the aortic valve, be shoved up through the aorta. I'm sorry, when they come through the left atrium, this is going to be oxygenated blood. And the reason that we get confused with this is because veins that have to do with pulmonary, this is opposite. So arteries carry CO2-laden blood, veins carry O2-laden blood. So the pulmonary veins are going to bring oxygenated blood back to the left atrium, through the AV valve, through the left ventricle, through the aortic valve, up to the top of the heart, through the aorta, through the arteries to take it through the body, down to the capillary bed. Capillaries and venules are going to exchange their gases, okay? O2 is going to go into the cells, and CO2 is going to go into the red blood cells. Those red blood cells are going to return via the vena cava, left and right, into the right atrium, through the right AV valve, through the right ventricle, through the pulmonary valve. And then that goes back to the pulmonary trunk, dumps it into the arteries. Again, CO2, only place in the body, CO2 is carried in arteries, is in the pulmonary trunk. Only place that oxygenated blood is carried in a vein is in the pulmonary trunk. This slide was talking about the basic structure of a blood vessel, and I wanted to give a comparison contrast in this slide to show you a large vein, which is shown on the top left, a large artery, which is shown on the top right. And when you look at the two of them, what is the main difference that you see? The main difference I see when I'm looking at them is the size of the lumen. A vein has a very wide lumen, and an artery has a much smaller lumen. I notice that the elastic layers in an artery are much greater than in a vein. And I notice that there's a lot more smooth muscle involved in an artery than there is in a vein. Both of them have low resistance to blood moving through them because they have endothelial cells that line the inside of the tube. But the size of a vein allows more to pass through at any given moment than an artery does. So and then you can see an arterial, a venule, and a capillary. And so they just get simpler and simpler the closer to the capillary bed that you get. And that should make sense because you've got O2 and CO2 that has to move through the wall of that capillary vessel. And if it were as thick as this artery or as thick as this vein, there's no way that CO2 would be able to diffuse out or O2 would be able to diffuse in or vice versa. It just wouldn't happen. So as the blood flows from the ventricles through the capillary beds to the atria, the pressure decreases in the blood, because the total cross-sectional area of the capillaries increases dramatically, leading to a significant increase in resistance to blood flow and a corresponding decrease in pressure. So pressures go down as the blood flows from the ventricles through the capillary beds. So from the heart through the systemic system down to that micro area that we talked about in this original picture that looked like this, down to that micro area, your pressures are decreasing, okay? Because there is a resistance to blood flow in those capillaries, because there's so many more of them. They're much smaller and much not as wide, and so there's an increased resistance to the blood flowing through there, which causes a decrease in pressure. Arteries. So let's take a look at arteries in a little bit more detail. So blood that enters an artery from the heart, a measurement. So here we're looking at what is this? This is our artery, okay? And then we turn into arterioles, which in our previous picture shows that they get smaller. Okay, so that's what this is indicating on this end of the picture. Okay, arteries are a little bit bigger. Okay, going back to this is a valve. Okay, so systole is measured as the blood moves through that valve, increases the pressure as it passes through. It's going to increase the pressure on the walls of the artery because they are elastic tubes. In addition, they have a low resistance to that pressure because they have a large radii, or a large space inside. Okay? Now, we look at it during diastole, when no more blood is coming into that particular artery. Now we have forces that are pressing down on the walls, and because they are elastic, there's low resistance, and that keeps the blood moving into the arterioles. Okay, so blown up, filled up, resistance on the outside, but because it's elastic, it allows that blood to move through. Once the blood moves through, because the pulmonary valve or aortic valve has closed, then you're going to get a reduction in size, but that's going to serve as a pressure reserve because it maintains the push of that blood all the way out by its elastic recoil. So you can imagine that the force is now pushing down on the sides of that artery, and any red blood cells, plasma, platelets, whatever else is in that blood, that's still in here is going to get shoved or pressurized out into the arteriole. So that's what keeps everything moving in the right direction in an artery. So how do we use the arteries to tell human health? Um, Pulse pressure is measured in the arteries of the wrist, and it is the difference between the two pictures in the slide that we showed before, the extended arterial or artery and the recoiled artery. So that's what's going to give you that pulse sound that you feel, the ba-bump, 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 ba-bump. It's that elastic recoil, fill up, recoil, kind of like a balloon. Then it fills up again, recoils, fills up again, recoils. And you're feeling that in the arteries of the wrist. Arteriosclerosis can be detected in the age population due to a decrease in compliance of the arterial wall. So what you're going to see is changes in pulse pressure in geriatric patients or the geriatric population, because as we age, all of our muscle cells, which have worked so hard, so long, doing their job for us, are now not as elastic as they used to be. And as a result, that pressure reserve that we got from the compliance of the artery walls during diastole is no longer there. And so a lot of times we're going to be able, arteriosclerosis then is blockages or a change in that pulse pressure. So think about on this slide, and I'm not going to say a lot about it because I think you can read it, and I also think that you've had enough experience with this that you'll be okay with it. But how do we use the pressures and elasticity of these vessels, arteries, to determine our blood pressure? Okay. And I'll let you look at the SFIG and determine by, so kind of spend a minute here making sure that you understand, you know about the sounds, what you're supposed to be listening for as you go, but connect it with what's actually happening inside of the artery and take a look at that. So what controls blood pressure? If blood pressure is a necessary measurement of the amount of force that is necessary to keep blood moving in this closed system, what is it that controls that? And that is a nervous system control. So baroreceptors, which are nervous system endings, are attached to arterioles, carotids, and the aortic artery. And so that information then can be relayed to the medullary part of the brain, the base of the brain, and that input then is sent into a very specific cell spot, set of cells in the medullary space in the brain. And as a result of that information, if the blood pressure is too high, if the blood pressure is too low, based on the signal that the medullary system is getting, you may have an increase in parasympathetic outflow to the heart. You may have a decrease in sympathetic outflow. You may have, therefore, a decrease in cardiac output. You may have a lower BP. Sorry, that should say higher BP. This should say higher BP. The opposite would be true if you had a blood pressure decrease. So instead of the input saying to the medullary cavity, we need to slow this person down. So this is a fight or flight situation. If the opposite were true, then you're going to lower the blood pressure by inputting into the medullary system. That's sending information to give a parasympathetic low response and an increased sympathetic response, so that you have an increase in cardiac output, and so that you have a decrease in cardiac output and you have a decrease in blood pressure. So the control of blood pressure is baroreceptors, which are nervous system cells that are directly attached or innervate arterioles, carotid arteries, and aortic artery throughout the body. So the changes that can be picked up by the receptors can be affected by a lot of different things. So if a baroreceptor picks up on a blood volume change, okay, you have a wound and you are bleeding profusely from the wound, those baroreceptors are going to pick up or signal that the blood volume that's passing through the artery is not normal, and it's going to send information to the medullary cavity in the brain, so that some sort of response. Would you want to increase your blood pressure there, or would you want to decrease your blood pressure? You'd want to decrease it to maintain as much or minimize as much blood loss as possible. If you have a change in kidney function, because the kidneys are putting out nutrients, ions, all different kinds of things into the blood, if you are worried about that, then those baroreceptors can say, hey, wait a minute, this level of a nutrient is off. We got to do something about this. Let's get it changed. Blood gas levels, pain, emotions, sexual activity, elevated intracranial pressure, that's in Cushing's syndrome, or part of the Cushing's response, will all cause those baroreceptors to send signals to deal with either an electrical response or a hormonal response or a combination of both, so that target cells can make responses that will minimize pain, that will try to balance out blood gas levels, that will try to minimize painful emotions, that will elevate any kinds of emotions that are associated and necessary for sexual activity, etc. So here, this picture is just referring to 3 different things that are caused when you have intracranial pressure, usually due to an accident. It's going to cause a change in your heart rate, it's going to cause a change in your breathing rate, and it's going to cause hypertension. And if you see all 3 things in a person that are sudden onset, it's partly going to be used to diagnose an intracranial leak or abnormality. Critical hypertension syndrome symptoms: persistent anxiety or agitation, tissue swelling, and shortness of breath. Okay, I show this slide because, again, hypertension, so hyper means above, so blood pressure that is too high, is associated with persistent anxiety or agitation. So one of the symptoms of a critically high hypertensive syndrome, somebody that's presenting in the hospital with a critically high blood pressure, is going to be that persistent anxiety or agitation. And based on what we just talked about, that baroreceptor response, That should make sense to you. Okay, that you're responding in a fight or flight way due to nervous system signals. Tissue swelling due to capillary leakage is going to be the lymphatic system connection, but that's going to be because of the incredibly high pressure in these very, very thin-walled capillaries. So there's going to be leakage of material from inside those thin um, layered capillaries out into the interstitial space. That is taken up by the lymphatic system, which we'll talk about in one of the next chapters, and then that leads to the swelling. And then that swelling can also cause fluid to build up in the lungs if the lymphatic system brings that fluid to the thoracic cavity. And there you have a respiratory system connection where shortness of breath as a symptom then can be linked back to hypertension. So these three things should be associated with a very, very high blood pressure and should probably make sense to you by now about why. So here's that process of blood loss that we talked about, where if you, for whatever reason, we talked about a needle stick, but for whatever reason, if you have loss of blood due to a vessel and injury, so again, you could have this kind of leakage from an accident. You could have this kind of leakage from the hypertensive syndrome that we just talked about. You can have it from degeneration of the walls for various reasons of the capillaries. But in any case, this process that we talked about early on is the actual coagulation process. But we didn't talk about the fact that the actual vessel is involved in making that platelet plug. And that is, there is a vascular spasm, which will move materials. Okay, so it's going to increase the pressure within the capillary to move materials past that break. Okay, and to get the platelets to the break to make the plug. So red blood cells are pushed past with that spasm. Platelets are brought along with it, but those platelets are dropped off and drawn into chemically that hole to make the plug. And then your fibrin filaments come in and make the net, and that's where the coagulation occurs, and it will close around that capillary. Any blood that accumulates, if these capillaries are not deep and they are on the surface, any blood that accumulates in the tissues around the wound is called the hematoma or bruise. And so when you're in this first stage, and maybe even in the beginning of this vascular spasm, you're going to have red blood cells and blood material that leaks out into the interstitial space. And that's what shows up on the surface of your body, and you can see it. Massive arterial blood loss is another issue altogether. And although we called the last slide hemostasis, returning a capillary to normal, now we're talking about hemorrhage. We're talking about massive blood loss, which will alter the blood gases because you're going to signal to the body that you have a lower oxygen level because you have fewer red blood cells. So that's going to cause a chemoreceptor reflex. And then you have decreased arterial pressure, okay, because that artery is opened up to the outside, so the pressure goes down outside as the blood as the blood flow diminishes because you're losing it to the outside of the body. And that's a baroreceptor response from the nervous system. And both of these things together are going to stimulate the heart in such a way, stimulate vasoconstriction in such a way, and stimulate volume redistribution of blood in such a way to minimize the massive arterial blood loss until an external process can be used to treat or close the wound, whatever that happens to be. So you're putting pressure on that thing to help with volume redistribution, which is already being done by the body. You're putting pressure on that to help with vasoconstriction, um, which is already being done by the body. And there's cardiac stimulation that's being done to reduce the amount of blood flow that is going to that particular area until that wound is closed. So what that is actually doing, that response, that nervous system response is actually doing, is autotransfusion at the capillary level to increase blood volume there and defer it from those huge arteries or veins that are bleeding out. Hypotension describes the issue of low arterial blood pressure. Um, it's started by the vasovagal, which is a nerve that runs from the side of your face down to your neck. The nervous system response that decreases sympathetic response and increases parasympathetic response. So ultimately, and we talked about that in the last chapter and a couple of chapters before in the nervous system, ultimately you want to reduce the heart rate and the blood pressure um, so that the blood is not going to be pumped at a very, very high rate out of that wound. The problem with that is if you are still losing blood, even if you decrease that, okay, so you're decreasing it, you're sending it somewhere else, the oxygen level of your blood now is already low because you're losing blood flow, and now you're increasing your heart rate and your blood pressure, so the oxygen that you do have on your red blood cells is not getting to your brain as efficiently as it was. And that's what can lead to the unconsciousness that's often associated with a massive hemorrhage. The next thing we're going to take a quick look at is arterioles. So that's the next level. Arteries leading to individual organs. Collectively, the term that we use for arterial blood pressure is MAP. Their diameter is controlled by three different systems. So the nervous system can control their vasoconstriction or their vasodilation. Hormones can control whether an arteriole is wide open or closed, not completely closed, but wide open or narrowed, I should say. And then there are also some local chemicals when you need an immediate response that can also constrict or dilate the blood vessels to respond to any kind of an issue that might be going on in the body. So major factors that we're talking about, so the neural controls that we're talking about are norepinephrine. Remember, that is a neurotransmitter, also a hormone. And then nitric oxide, which is also a neurotransmitter, and both of those can act on the smooth muscle that surrounds that areolar radius that we saw back in the previous picture. They alter that smooth muscle to either expand or contract. Hormonal controls would be epinephrine, angiostensin 2, vasopressin. We talked about that in the last chapter. Those can act as vasoconstrictors, and epinephrine shows up on the vasodilators list as well, which might seem kind of weird, but remember we talked about concentration levels. That's what we're talking about here. And then local controls. So a decrease of oxygen is going to automatically cause a vasodilation. A decrease in potassium, carbon dioxide, the presence of potassium, CO2, and hydrogen is going to cause vasodilation. Amount of water in the plasma, changes in that could cause vasodilation. And then all of these other substances or chemicals can also do that. But in any case, all three of them. So these are the chemicals. This is the type of control. And this is what we're acting on, the smooth muscle in the outside of the arteriole. Work our way down to capillaries. It's the major site of diffusion. I love this picture. It shows a capillary being only as wide as one red blood cell. So this is a red blood cell on its side. That's how wide a capillary is. Sometimes it can be a little bit bigger, but they permeate every tissue in your body except the cornea. Not necessary for the cornea. That is where gas and nutrient exchange occurs with the fluid that surrounds the capillaries in the tissue, so that all of the gases and nutrients can get into the cells that need it for cellular respiration. You're exchanging not only CO2 and O2, but lipids, ions, polar molecules, proteins, water. Whether that kind of diffusion is active or passive or facilitated, and these are terms that you should already know from a very, very early chapter when we talked about movement in and out of a cell. Active requires energy. Passive does not require energy. Facilitated does not require energy, but it requires help in the form of proteins in the cell wall that will allow materials to move in when the cell wall itself will not allow materials to diffuse through because they're too large or they're polar or non-polar or whatever it happens to be. Diffusion is usually from a high to low concentration. So that goes back to what I was talking about when I said, okay, in arterial blood, oxygen pressure is high on the red blood cells themselves. It's low in the interstitial space because the oxygen that used to be there was used up by the cells. And so the oxygen is dropped off and the CO2 levels. And when I say it's dropped off, it's diffused. The oxygen that would be on this red blood cell is diffused out into the interstitial space to be taken up by cells. Hydrostatic pressure, and we can use the prefix hydro because water is involved, huge component, 90% of the plasma of the blood. Hydrostatic pressure favors movement out of the capillary into the interstitial fluid, into the lymph. Okay, just the fact that there's more water here than there is in the interstitial space would cause water to want to move out of here into the interstitial space. And when it goes into the interstitial space, it becomes lymph. Oncotic pressure, on the other hand, is protein pressure that causes movement into. So there are more proteins here. There are more proteins inside the cell than there are outside the cell. So all of the other little chemicals want to move inside the cell as a result. Structure number 4, veins. One of the main differences that we saw with a vein is that it had a very wide lumen. Also that it was not as muscular as a artery. It has a high compliance. That means that it's able to move material through it because it has thinner walls. It has a very low resistance because of the endothelial cells inside of the vein. that cause a smooth surface for the blood cells to be able to pass through very quickly. Sympathetic control can either dilate or constrict it because they do have smooth muscles, not as many as the arteries, but they do have smooth muscles. Skeletal muscle that surrounds the vessels can help pump blood and increase venous pressure, which is extremely necessary because veins are often moving against gravity. And so instead of the arterial blood, which is moving with gravity away from the heart, venous blood is moving against gravity. And so the skeletal muscles that surround the outside of these veins oftentimes will squeeze the walls, so tiny squeezes that will pump it through. And then these valves will keep the blood from being pulled back down on heartbeats. Okay, so if blood moves this way and then there's a release from the heartbeat, and then it would move again, and then it would keep getting pumped up, keep getting pumped up. Now you can see, so it would be start, stop, start, stop, start, stop. And if it's against gravity, now if you're going with an artery and it's start, stop, doesn't matter. Start, stop, start, stop. It's still going to constantly move in a downward position. But you don't want that with veins because you're trying to get back up to the heart. So these valves are going to keep it from backflowing. So go through the valve, hit the valve, the closed valve. Go through skeletal muscles, move it further. Hit a valve, go through a valve, continue that process all the way through. If the vein valves are not working properly, you can end up with varicose veins. And that's where you get pooling of blood clots and expansions in those swellings in those veins. Very, very painful because they are blockages. And it does not help with the movement, the efficient movement of blood back up to the heart. The blood being pumped through the heart chambers does not exchange nutrients and metabolic end products with the myocardial cells. So I remember learning about the cardiovascular system and thinking, oh my gosh, I didn't even think about the fact that the heart is an organ and it needs its own blood supply because the myocardial cells have to have oxygen, ATP, to do their sliding filament thing and be able to work. And so where are they going to get their own blood supply? So there are arteries attached to the backside of the backside and sides of the heart that actually supply the muscle cells themselves with oxygen and take away CO2. The coronary arteries actually exit from behind the aortic valve cusps in the very first part of the aorta. And most of the cardiac veins drain into a single large vein from the heart with excess CO2 as a result of cellular respiration inside of the cardiac cells. Okay. And that coronary sinus dumps right into the right atrium. Okay, a cardiac angiography or the balloon procedure can access the coronary artery openness and blood flow. So when we're looking at a blocked artery, when we're talking about open heart surgery, when we're talking about the angioplasty, the balloon, we're talking about looking at these arteries for blood clots or plaques. When a coronary artery becomes blocked with a plaque, and that's usually a fatty substance that's put into the vessel, and it has to do with your diet. As dietary sometimes can be made worse by genetics, you get atherosclerosis. So those plaques, because the heart is not getting the oxygen needed to do the work of pumping blood, again, it's a cascade effect. If your heart can't pump the blood, your body cells can't get the oxygen. They can't dump off the CO2. They can't reoxygenate through the respiratory system, and the whole body system breaks down. So that's why heart attacks are such a big deal. These coronary arteries are necessary for the health of the cells that are responsible for pumping the blood to the rest of the body. Distribution of systemic blood flow to the various organs and tissues of the body at rest. Kind of wanted you to just see where blood flow is going in your body in terms of and how fast it's traveling. So 650 milliliters per minute is the travel rate in the brain. Compare that with skeletal muscle. Compare that with abdominal organs. Okay. This is the only thing this is really good for is to just get you to think about what requires a lot of blood and why. So does the brain require as much blood to function as skeletal muscle? And why? Does the skin require as much blood, knowing what the job of blood is? Does the skin require as much blood as skeletal muscle? And why? Why do the abdominal organs require so much flow per minute? Why would that be? Think about the answers to those questions in terms of what you already know about the nervous system and the muscle system. I don't know if you guys, I know you covered integumentary system and the digestive system a little bit in Anatomy 101 and 102, but just think about what they do. And now that you know what blood does for the body, think about what they would need. So how do you determine the flow rate? It's physics, a change of pressure divided by a change of resistance. It's very, very similar, although not exact. It's very similar to what we look at in electrical flow. There is a difference because a blood vessel has a lumen and a copper wire does not. But blood flows from a region of high to low pressure. Resistance is a measure of the friction that impedes the flow through the blood vessels. So the change of pressure has a direct relationship with the flow rate. Okay, and what's going to cause the change in pressure? We talked about all of those things that can change your blood pressure. There's a direct relationship then. When your blood pressure changes, the flow rate changes. If the flow rate changes, relate that back to the table on the previous slide, and you start to see the cascade of issues that can happen. There is an indirect relationship between flow rate and resistance. So what that means is as the resistance of the vessel goes up, the flow rate goes down. So that would be negative, would be a bad thing. You don't want the flow rate to go down unless you have an open wound. Okay, and maybe you want to change the resistance in a local artery because you don't want to bleed out. So in that case, a higher resistance would be positive because it would slow the flow rate past that wound opening and allow for less blood loss. Viscosity of blood, the length of the blood vessel, how far the blood has to travel, and the blood vessel radius then would all be things that can impede flow rate. this should very well be just absolutely second nature to you at this point. The parts of the heart, the anatomy of the heart should be second nature to you. The one thing I want to point out about this is that discussion. I want to mention one more time that discussion about all arteries carry oxygenated blood away from the heart, except the pulmonary arteries, which carry deoxygenated blood to the left side of the heart. Because remember, pulmonary arteries are working with the deoxygenated side of the heart, where blood has returned to the heart to be processed in the lungs. It's the only place. Don't ask me why. I don't know why they did it. I know there's a reason. You could probably look that up pretty easily, but I don't know that reason. Also, all veins in the body carry deoxygenated blood. So all arteries carry oxygenated, all veins carry deoxygenated. The exception is the pulmonary veins. Notice that they're associated with the left side of the heart. And the left side of the heart deals with oxygenated blood. We know that because the aorta is responsible for taking blood to all parts of the body. A reminder that the heart wall is made up of epicardium, myocardium, and endocardium, 3 layers that allow for the efficient and long-term pumping of the cardiac muscle cells. So epicardium is protective but elastic. Myocardium is the actual structure of cardiac muscle. And then the endocardium is this lining on the inside of the ventricles and the atria that allows for blood to flow through and move to the different areas that it needs to move through. Movement between the atria and the ventricles and those tubes that are taking blood away from the heart, those are all monitored or separated by valves. And you should have those memorized by now. So what we're looking at on this picture, we're looking down into the heart as if we've cut the top off, we've pulled the lid off, and we've looked down into it. And this is what we would see. The semilunar valve, the aortic valve, and openings to those coronary sinuses or coronary arteries that we were talking about. The bicuspid valve and the tricuspid valve. How do you know the difference? 1, 2, 3 is tri. 1, 2 is bi. So true or false, cardiac muscles of the myocardium are arranged in layers that are tightly bound together. That is true, but they have gap junctions in between them that allow for a little bit of communication. The atria contract together, followed by the ventricles contracting. And I talked about in lab, I like to call this ka-chunk. So both atria would represent the ka part of the heartbeat, and both ventricle pushing blood up and out of the heart are the chunk part. Almost every heart cell contracts with every beat of the heart. The cardiac muscle beats 300,000 times in your lifetime. What do you think about that? True or false? Absolutely false. Human heart cells are replaced weekly. True or false? What do you think about that? That one is true. Not all of them are replaced in a week, but some human heart cells are replaced weekly. So the electrical system of the heart is also directly attached to the nervous system, because anything that has electrical conductivity in it is going to travel on CNS or PNS nerves. So parasympathetic and sympathetic nerves attach to heart muscle cells via gap junctions. So that's what I was talking about before. So this is a skeletal-muscular connection. You've already seen that one. So that's where a nerve comes into contact with a muscle cell. Okay, so a skeletal muscle cell. That's not what we're talking about here. The nerve here is going to come into contact with a cardio muscle cell, and there are numerous little gaps between the two. That's where the information is passed through. Whereas here you had a synaptic connection, okay? You had a synaptic space. Here you have gap junctions. And in succession, so that nerve electrical signal action potential is going to travel through here, and at the gap junction it's going to be turned into a chemical response that causes these to contract. Here you had a action potential, traveled through the nerve, comes to the ending, and it turns into a chemical response. The chemical causes each of the cells to respond, muscle cells to respond. Hormones involved in the response that we were just talking about are norepinephrine and acetylcholine. And we've heard about acetylcholine before because that has to do with the acetylcholine has to do with skeletal muscle contraction as well, but it's also used here. So norepinephrine can control heart rate, blood vessel dilation, and muscle relaxation. Epinephrine from the adrenal glands can also work on these receptors in the same way, but epinephrine is going to respond to different signals. Because it's from the adrenal gland, it's going to respond to different signals in the blood than norepinephrine, which is going to take those baroreceptor signals that were sent up to the medullary part of the brain and do a different type of reaction. Whereas epinephrine is going to be coming from the bottom part of the body, your adrenal glands, on your kidneys, and then it's going to have some sort of an effect on the heart, the blood vessel dilation, or muscle relaxation. Acetylcholine is parasympathetic, and it's going to help with the rest and relax response versus the elevated fight or flight response. So parasympathetic responses are via vagus nerves. Those are the ones that run down the side of your face and into your neck. So an electrical signal is going to come down here. It's going to stimulate acetylcholine production that's going to hit muscle cells via gap junctions. It's going to hit muscle cells of the atria for a response. Okay? If it's parasympathetic, is it elevating a response or is it diminishing a response? Think about that. So the opposite then, sympathetic, is where you're taking thoracic spinal nerves. So we're not sending anything from the brain. Thoracic means from your chest area, coming out of your spine. These nerves are going to go to the atria, and they're going to stimulate norepinephrine release on different types of receptors through the gap junctions, by the way, not a synaptic capsule, not a synaptic space, but through the gap junctions. They're going to stimulate atrial muscle cells using a different type of receptor. Epinephrine sent from the adrenal glands, okay, via the bloodstream, because it's a hormone, so this is a hormone response. They're going to be delivered to the blood ventricles, again, passing through and attaching to receptors on the gap junctions so that the ventricles will respond in a sympathetic way. So sympathetic is fight or flight, parasympathetic is relax and rest. So what do you think are going to happen to the atria when acetylcholine acts on them? Are they going to contract and contract faster, or are they going to slow down? When norepinephrine reacts or acts on the atria, are they going to speed up contractions of the atria and therefore heart rate and blood flow, or slow it down? Action potentials from nerves in all heart muscles have rapid depolarization compared to any other muscle cell type that we have. And that's primarily related to increased sodium permeability in the muscle cells, the cardiac muscle cells themselves. So cardiac muscle cells are more susceptible and more capable of taking in sodium to get the process of depolarization started. Okay, so in other words, once you have a neural response, parasympathetic or sympathetic, it's going to cause a rapid contraction of cell muscles or response in cell muscles, much more so than it would if it were having to stimulate skeletal muscles. It's a much faster process. Cells also remain depolarized for the duration of the contraction. So the polarization, so it's a very strong contraction. And you can imagine that it would need to be because of the force necessary to move the blood through the heart. Now that is due to L-type Ca calcium channels. Most other muscle cells do not have this particular type of channel. So larger, stronger calcium connections, increased sodium permeability, all of those things together cause a really, really fast response to a nerve stimulation. Heart pumping. Hopefully this is review, so we'll go through this pretty quickly. The SA node sets the pace of the heartbeat. The action potential goes through the myocardium via gap junctions. Depolarization through the atria cause them to contract at the same time. That's the K sound. There is a 0.1 second delay through the AV node. So here's your SA node, here's your AV node. So in here, we're getting depolarization through the atria. And when you depolarize, you contract. When you contract atria, you push blood down into the ventricles. Both ventricles at the same time, because both atria, and you can see this here, both atria are innervated with nerve system action potential that causes them to contract or get smaller. Push that blood through those valves, and then there is a 0.1 second delay, so the AV node slows it by 0.1 second to prevent tetanic contractions. Okay, so it allows the atria to relax before they try to start pulling blood in again when the ventricles haven't even released their blood yet. So the whole heart would start going blah, 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 because you've got blood from one contraction up here pushing down here. If there wasn't a delay so that the ventricles can depolarize to move their blood out of the heart, you would have more blood coming down in here. So you would have a ka-chunka, 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 which would not be healthy for the heart. An AV conduction disorder can be caused by drugs, different types of disease, but it can either reduce or eliminate the transmission of the action potentials. And when you do that, you obviously are not going to get a depolarization of the atria. They're not going to move the blood cells, the blood to the ventricles. The ventricles can be out of sync then, because if the AV node is not firing properly, it's not allowing the ventricles enough time to depolarize before the atria are going to shoot that blood down in there. So the current treatment for AV node conduction disorder is a pacemaker. And that pacemaker is going to stimulate the ventricular cells at a normal rate. So it kind of takes over for the bundle of his and says, okay, the pacemaker is going to regulate that delay so that these are able to contract in a timely manner. So the AV node sends an action potential after the delay to the bundle of his. The bundle of his will send that action potential out into both branches of the heart, and the ventricles will contract at the same time. And that's what I always call the chunk. And so they're moving their blood out into the body by depolarizing. So another issue with blood flow within the heart is going to be a valve dysfunction then. And normally blood flow through the valves is very smooth, very concentric when you're listening to them. What you're going to hear with somebody that has a valve dysfunction is turbulent flow. So it might show stenosis, that there is a narrowed valve, and that can be from calcium buildup, it can be from fatty buildup on the valve itself, so that blood is not going the direction that it's supposed to go in an efficient manner. That's stenosis. You can get blood that flows backwards through a leaky valve. That's called insufficiency. And then you can get blood flow between the two atria or two ventricles through a hole in the actual wall that separates them, the interventricular space or muscle. And that's called a septal defect. And you're going to hear all of those as turbulent flow. So normally flowing blood is going to sound smooth. You're going to be able to hear the heart rate, but it's going to sound smooth. Turbulent flow is going to sound like an interruption. We've talked about this picture in lab, so I'm going to let that go. But you should be able to tell both on the exam for this chapter and the lab practicum that you're going to have where the blood is in each of these scenarios. So when you're talking about the P wave, where's the blood in the heart? When you're talking about the QRS complex, where's the blood in the heart? When you're talking about the T wave, where's the blood in the heart, etc. So P wave equals atrial depolarization, and we said that when something is depolarizing, it's contracting. So if both atria are contracting, during the P wave, blood is being ejected into the ventricles. During the QRS, you're experiencing, so you've got blood in the ventricles, and the ventricles are depolarizing, which means that they are pumping blood out of the ventricles and into the body. And the T wave indicates ventricle relaxation. So it is that point before the SA node begins its stimulation and depolarization of the atria, to pump the blood back into the ventricles and start that process all over again. This picture is associating systole and diastole with each of the movements of blood in the heart. It's also associating those with the complexes that are associated on the EKG. So as you can see, the ECG is used to measure the electrical activity that causes the heart to pump. It's not a good measure of the activity of the valves within the heart. You would use an echocardiography for that. So it's a picture of heart valve movements done with an ultrasound. Okay, so this is a better measure of movement of blood within the heart. But if you want to get a picture of what is the turbulent sound that you might be hearing from the outside of the person when you're diagnosing a heart issue, you're going to want to use a different test for that and get a picture of those valves to see whether it's a calcium buildup, to see whether it's a torn valve, whether it's a torn tricuspid, whatever it happens to be. I do want you to watch this video, so please, it's extremely important that you watch this video. I'm not going to show it on here because it's a waste of time, and this file is already going to be pretty big, so I want you to watch this one on your own and take notes. Its focus is pressures within the heart and how pressure changes cause blood to move through the heart. So it kind of brings it all together for you, the cardiac cycle. So please get the live version of this PowerPoint from Module 10 in our classroom and watch it. It's within the PowerPoint live version. Arrhythmias we spoke about in class. I'm going to let that go. The only thing I will say about arrhythmias is that a lot of times potassium levels are involved with repolarization, getting the heart ready to do the next heartbeat. And so if you have an excess or a depletion of potassium ions in your diet, in your body, accessible by the heart cells, you may see arrhythmias on the ECG. So an important connection to arrhythmias is potassium availability. Cardiac output is a product of the heart rate and the stroke volume. Normal cardiac output is about 5 liters per minute, meaning that your heart can process 5 liters of blood per minute. Okay, that would be normal. So it's the volume of blood pumped out of each ventricle per unit of time. So that's a lot of blood. Factors that can change your cardiac output are chemical. Epinephrine can cause an increase in cardiac output. Catecholaminergic action, those are also chemicals, causes an increase in cardiac output. And cardiac output is also increased cardiac output will be decreased when the afterload of the heart is increased. So if the heart is working really hard to eject blood, that is its afterload. How hard it has to work to eject the blood is its afterload. An increased afterload reduces the contraction. So it's kind of an energy exchange issue. Okay, if there is an increased afterload or an increased requirement for energy to eject the blood for whatever reason, exercise, etc., there's going to be a reduced contraction to compensate for that energy loss. Factors that change your heart rate. Parasympathetic intervention can decrease the heart rate. Sympathetic can increase it. There's a genetic control because of the nerve intervention to the heart. Um, Genetics plays a part in how many nerves are attached to your heart to cause it to have a normal heart rate at rest. And in fact, while some of us can have a resting heart rate of 60, 68, 70, some of us sit at a heart rate, a resting heart rate of 90, 95. And that might be under genetic control because of the differences in nerve intervention that we have in our heart. And the SA node pacemaker, its efficiency and its ability to work are also factors in controlling heart rate. Why do we exercise? We exercise because we are trying to balance the efficiency of the heart. As we talked about before, we want to increase cardiac output to the muscles because typically if your afterload is high, you're going to decrease cardiac output, right? The stronger your heart is, the lower the afterload is. The less difficult it is for it to increase the cardiac output without using up your energy. So the more you use your heart, the stronger it gets, the lower the afterload, the higher the cardiac output to the muscles, and the stronger you are. Remember, cardiac output is going to increase oxygen movement to your muscles, and therefore it's going to increase the muscle efficiency in response to whatever exercise you're doing. Increased blood flow also goes to the skin for heat release. So it helps if you have that higher cardiac output, you're able to release heat more efficiently and therefore keep the afterload down so the cardiac output can stay up. It also goes to the brain for nervous system support of heart and muscle function, and it diverts blood away from the digestive system while you're working out so that your body is burning fat or different sources of energy and not the food that's in your stomach. So you can actually, your digestive system gets a chance to relax, gets a chance to not have to work as hard, and your heart gets a workout so that it can work more efficiently. Homeostatic imbalances, there's so many of them. I'm hoping some of you might pick some of these um, for your topic. I don't remember if any of you did, but ischemia is the general term for inadequate blood flow in the closed system. There are all kinds of ways that that can happen, okay? And you can look up any one of these. You looked up some of them and discussed some of them in your cardiac case study, so I'm not going to go into each one of these, but I want you to take a look at them and think about what the words mean. Myocardio, so myo refers to muscle, cardio refers to cardiac. So break some of these words down using your your medical terminology class. Think about what hyper means, what trophic means, myo again, cardio, apathy, um, and try to work some of these out. It'd be a great place to practice um, that med terminology.